This meeting is being recorded.
Hello, everybody. Welcome to our meeting devoted to CAFA challenge. So this is a challenge on the Kaggle platform to predict properties of the proteins from their sequences. Today's talk will be by Liza Geraseva, who will give overview of the methods used on the previous CAFA challenges. Thank you very much, Liza, that you agreed to make a talk. So, colleagues, we are very informal, and so don't hesitate to ask the questions during the presentation, but please write them in the chat, and then I will speak out them. Okay, Liza, thank you very much, and please start.
Good evening. I am going to review some of the methods used in CAFA competition number 3 in previous years. So here is the graphics of the results of CAFA 3 competition, and I'm going to review some of these methods, concentrating first of all on methods obtained with this laboratory and in this laboratory, and I'll also want to introduce methods obtained by Hoehndorf lab, because they are at the top of the leaderboard on CAFA 5 now. Well, first of all, I'd like to introduce some baseline methods. Most baseline methods in CAFA competition are based on blast search of similar sequences. So, and there are 2 of them. The idea is to use the normalized bit score obtained by blast method and blast like diamond and so on. And the idea is to normalize the bit score with a positive prediction label on the maximum bit score obtained by from the similar sequences. And another baseline method is to normalize by the sum of the bit scores of similar sequences. And I have tested both methods on CAFA 5, and I obtained these results. So these methods are baseline, but though they are rather effective. So, and in these methods, the program used to obtain similar sequences for every query sequence from the test set is a method called diamond. It's a blast-like method, and it also uses a seed and extend paradigm when exact occurrence of short words contained in query sequences are extended using Smith-Waterman algorithm. And there are some features in Diamond program that make this program more effective and fast, such as so-called spaced seeds, when you use the words with only longer words in which only some of letters are known, and they also use a reduced algorithm, and it shows that it was shown that these features doesn't reduce the accuracy of the algorithm very bad. So, and the latest Diamond version has several levels of sensitivity with different seed weights and shapes, and well, it's not interesting. Let's go to the methods used in CAFA 3, and so first of all, I'd like to tell about methods developed by Hoehndorf lab, and these are DeepGo and DeepGo Plus. And these approaches use convolutional neural networks on sequence data, and combine these with some external data. In DeepGo, they use also embeddings generated from protein-protein interaction networks from the string database. And well, you can see the you can see the architecture, the picture. As you can see, they use programs, then embedding layer, then convolutional layer, and then they use a hierarchical fully connected layer to generate predictions, so that as we know, the Go terms are organized in a hierarchical manner. So for each parent, the prediction mustn't be lower than the maximum predictions of all of his children. So this hierarchical classification layout is to overcome this problem. So, but the drawback of this layout is that it has a rather complex organization. So it was trained for a subset of about 2,000 Go terms. So we cannot use cannot train it for all the Go terms, about 40,000. Because it's computationally expensive. Well, and I've trained this model, and the public score on CAFA 5 is, you can see the picture at the slide. What the problem is that they use out-of-date embeddings from protein-protein interaction. So I couldn't how to say, I couldn't update them for the latest data set used in CAFA 5. So the score is so low because of this, I think. And another drawback is short protein length and a small subset of predicted Go terms. But I think it will. You can improve the score if you combine it with other methods. So, and another approach developed by this lab is DeepGo Plus. And in this method, as you can see, they get rid of the hierarchical classification layer, because it's computationally expensive and complex. And they also get rid of 3 grams, and they use only one hot encoding layer instead of the embedding layer. And they used a set of different convolutional layers with different strides and kernels. So, and this helped it helped to improve the score, and it helped to obtain more features from the final layer. They used a larger number of class, and they also use a larger length of sequences, up to 2,000. And public score on CAFA 5 is, you can see at the slide. And and what should I say? They do not use this architecture alone, and they combined it with the baseline method, BlastKNN, I remember. So, and when using with the latest data set, it provides acceptable score. Well, and I'd like to to review other methods from other laboratories. For example, methods from JULAB. There are two methods, and on CAFA 3, this lab was the winner, and they provide two methods. One of the methods is deep text to go, and this approach combines classical methods on different data. They include sequence data and BLAST KNN. They include sequence data, and they find interpro features for each sequence, and they make logistic regression on it. And they also provide, for each sequence, they also find citation abstracts from Medline, and they also provide include text-based methods, and they also make a logistic regression on it, and the consensus provides the prediction. Well, as you can see, these methods use rather simple machine learning techniques, but they include multiple data. They include text data from the citations. They include data from interpro, and they include a simple baseline method. So this lab uses rather simple methods in another approach, GoLabor, which showed better results on CAFA 3. What they do, they include rather simple methods, including logistic regression on InPro, on Profet, on camera rate. They include also BLAST KNN and naive approach. And from these simple methods, they obtain the initial list of predictions, and they for each method they find top 30 predictions, and these top 30 predictions they take as an input for a ranking model lambda mat, and then provide a re-ranking of these predictions. And in fact, I couldn't understand whether they used top K ranks, or whether they used the relevance provided by this model. But anyway, this approach is rather effective, nevertheless. As you can see, this approach can be used not only with those simple methods. This approach can be also used with another methods, whatever you like, and it can be used also for ensembling and so on. Well, and I would also like to have a look on last method. This method was second on CAFA 3, and this approach is based on the composition of a query sequence into structural domains, and then on scanning them against known functional family models with known Go terms. And these HMM profiles can be found at CAFA site, and they also can be used in the current competition. Well, that's all. Блин, давно по-английски не говорила.
Okay, Liza, thank you very much for your presentation. So now, colleagues, you're welcome. For just a moment, I'll allow you to unmute you. Yeah. So Liza, thank you very much again. And colleagues, please don't hesitate to ask questions or comments or suggestions or whatever.
Okay, Blastkanen can do better, as far as I know, like around 49. In fact, I tested it myself and I obtained this score, but maybe there are some nuances that can improve the score. Well, it's rather interesting. Well, there is also a neurosymbolic version of DeepGo for zero-shot learning, but also for exploiting the gene ontology axioms MHBenta that DeepGo or DeepGo Plus. It's a much improved version of the hierarchical classifier used in the original DeepGo. Oh, it's good. In fact, I like the idea of the hierarchical classifier because everyone uses some fully connected neural networks for to obtain the final predictions. But it's not. I think it's not a good idea because, in fact, Go term organization is hierarchical, and as I can see, only DeepGo uses this information. Well, I was upset when I found that in DeepGo Plus they don't use it, but if they do in another work, it's rather good.
Okay, colleagues.
I do have 2 questions. Hello.
Yes.
First is very basic. It seems like you described multiple methods from CAFA 3, but was the CAFA 4 challenge? And if it was, then what methods were used there?
In fact, I couldn't find any results from CAFA 4. I think they still analyze analyze the results because they as you can see. Well, where is it? Their full cycle of the CAFA 4 isn't still hasn't yet finished. So I saw that I didn't review the method.
Okay, I see, no problem. And the second question is also very practical. So you said that you have tested those models yourself. By that you mean that you used already trained models, right? So you
yes, I used pre-trained models, and I think if you make transfer learning of these models, it would include the score.
Okay.
But I trained only DeepGo and DeepGo Plus. So
just to be sure, I'm not that well familiar with the challenge, but there are different outputs that I see in terms of Go terms included. So how do they map on the expected outputs in the challenge?
I don't know quite understand what you mean. I mean that there is a large number of classes in the challenge, about 40,000. I mean, and both methods DeepGo and DeepGo Plus they cannot deal with such large number of classes, so they work only for a subset. So they are good for combining with other methods. In fact, DeepGo Plus is combined inside the approach. It is combined with another method, and it includes the score. But yes, it's used to predict only a subset of classes.
By the way, we have the author of the DeepGo, Robert Hoehndorf, here. So, Robert, if you can make some comments, suggestions, I don't know, you're very welcome. You can unmute yourself.
Yes, so thank you very much for this very nice presentation. I think it was very interesting, and it's true. I mean, everything you said about DeepGo is true. So DeepGo was a very big model. It was very difficult to train. It overfitted very, very easily. And this was also why the first results or the first model was very much overfit, and it failed to perform in CAFA. And this is why we had this very lightweight model, DeepGo Plus. But now we are using DeepGo Zero. So DeepGo Zero is our latest model. It has some hierarchical, not really hierarchical classification back, but our baseline model, or currently we're using our baseline, this as our baseline model. It gives around 0.6 in CAFA 5. After some tuning, so it needs a bit of tuning in terms of, so there is a big challenge in, so there's one big challenge about CAFA, and this is to balance between predicting for novel proteins, so proteins which are very dissimilar to proteins you find with some function annotation, and predicting for proteins which are rather similar. And usually we build our models so that they are performing quite well for very novel proteins. So there is a little bit of tuning that was required to get DeepGo Zero to work on CAFA 5, because there will be some sequence similarity between the proteins you predict for, and that will be used for evaluation, and proteins which are in the training set. And usually we build our models by removing the sequence similarity, but if we are too strict, then then we don't get a performance that is very good for CAFA 5. So this was the main outcome. So there's very little primary research yet in our models. So it's mainly a somewhat tuned DeepGo Zero as of now, with a little bit of modification. So you may want to also have a look at this paper and this model.
Thank you very much, Robert. Thank you very much. Okay. Colleagues, any other questions, suggestions to Liza or maybe to Robert? Or so. Okay, so.
Well, I would like to ask, obviously it's much better to just read the paper, but how exactly ontological axioms are used in DeepGo Zero? Is it some kind of knowledge graph embedding, or is it rather a symbolic approach?
Yeah, we use a neurosymbolic approach. There's a figure, if you go a little bit down, the figure shows that it essentially constructs a little bit further. It has a couple of circles in it. Maybe that's the easiest to explain this, but this one. Yes. So essentially, I mean, it's maybe not the easiest figure to read, but the idea is that on the right-hand side, there is what we call a geometric model of the gene ontology. It is essentially a geometric way to represent the axioms in the gene ontology. So hierarchy is just that circles are inside other circles, but there are also relations and quantifiers and disjointness, all part of this geometric space. And then DeepGo Zero, all it does is it projects a protein based on some embedding into the same space, and then infers the function by these geometric relations within this space. So this is the whole idea. And this space has all the axioms of Go in it. And on the left-hand side, you just take whatever representation of a protein you have, and you have a multi-layer perceptron, just a fully connected layer that projects or two, however many you want, but essentially just some very, very simple model that takes the dimension, that outputs the same dimension as you need in the space on the right.
Yeah, thank you.
Denise, if you have another question, please.
Just to you, basically, okay. Sorry.
Well, where we
Did that there is a question?
Okay. So, Alex, do you have another question, suggestions, comments around this talk, or maybe in general around the competition? So, especially those who are participating, like John or Max or Mikhail. So, if you have some question.
Can I address a question to Robert?
Yes, sure.
My question is, how do you use these new protein language models in terms of DeepGo 0? Are those the embeddings you refer to when you, for the protein embeddings? Is that what you are specifically talking about?
So there are two types of embedding. So essentially, looking at this figure, there is whatever representation of a protein you take. This is your protein P. So it could be an embedding. It can be some feature-based representation. And usually it has some size M. And the embeddings on the right, these are embeddings of Go. This is just gene ontology. And they usually have a different size N. And then there's all you need is multi-layer perceptron to take some representation of a protein of size N and map it into this space on the right-hand side of this figure of size N. So you can take any kind of representation of a protein that you like. So we did an experiment extensively in this paper with different protein presentations. We just used interpro features as an input. So this was all our representation.
Thank you.
So Robert, you don't use this T5 or ESM2 or whatever models?
We did not use it for this paper.
Okay, I see.
That says a lot. Yeah, thank you.
If I may ask, I didn't completely understand the fine-tuning part you referred to. So essentially, you removed some of the proteins to avoid this. Is this fine-tuning process what you referred to, or is it in the paper described in detail? Because I didn't completely get the idea. Robert, maybe you could explain.
I think it's about the tuning for CAFA challenge, but it's mainly when we train these models, we train on a split by sequence similarity. And the sequence similarity threshold by which we split training and testing may be very low. So it could be 30% or 50%. But this works well to generate a model that would generalize to proteins that are very different from any known proteins with a function. But it does not work so well for CAFA, because many of the proteins that are used for evaluation are actually quite similar to some of the proteins that we already know the functions for. So there is a bit of trial and error needed to get this to work.
Okay, thank you.
So yeah, just a comment. So I published a notebook on the Kaggle where I used this diamond, which was mentioned by Liza, and it has this several. You can choose the thresholds of the similarity, so you can obtain different cross-validation of test and train splits with different levels of similarity. So I don't know what Robert is choosing in their solution, but maybe someone can look at this notebook and try to also use it. But in a sense, that's a kind of a little bit dangerous, that if you look on the public leaderboard, you may be getting a good result. But if the private leaderboards will be different, then I don't. Yeah, that's a big question. How to choose this similarity threshold? Maybe it's very good for public, but who knows what will be on the private leaderboard? I don't know if someone has a suggestions or comments on this, it would be great. Okay, colleagues, any other questions, suggestions, or comments?
Sorry, can you describe metric? It was post about metric, and can you speak about this?
Liza, can you add a few words about metric? Or?
Well, about metric, I cannot say anything because it's rather difficult. Well, what should I say?
Okay, maybe someone can comment on the metric. I don't know. So my understanding of the metric is that, so first is similar to what is called F1 metric, which is quite well known in the data science, but in a sense, it's not exactly the standard F1. That should be understood. That, for example, you can use F1, standard F1, if you have only one target. But here, the computation is quite different. In a sense, so we have many targets. And what is done, we calculate precision and recall along the target axis. Then you make an average over samples, and then only you calculate the F1. So metric is pretty complicated for this competition, I would say. I don't know what is the opinion of the other participants, but there are comments from Predrag. We don't know different it will be.
Yeah, can I comment? I can provide a couple of comments on the metric just to briefly explain the rationale for it.
Yeah, sure. You're very welcome.
Yeah, you can think of this type of classification as multi-label classification, but it's more than that because certain terms are implied by other terms. So it's multi-label classification with dependencies, and certain nodes are easier to predict, or we think so, than other nodes. And because of that, we decided to value less the node, let's say the top node in the ontology, and so to score it as a 0 actually points, because every protein that has any function in, let's say, molecular function ontology will have that top term in the graph. And therefore, all the nodes are weighted based on information theoretic approach. And that's incorporated into the F measure. And so there's really, it's not that complicated. It's maybe that it's unusual for the communities that have not been predicting on these hierarchical representations. And so we just try to address this problem. Otherwise, you can get very high precision if you predict only the top term. And your recall will be small, but just by a couple of other terms, you can sort of find a good prediction balance by not doing really well biologically. And therefore, we value a little bit more those terms that are deeper down in the ontology. And that's what the metric is trying to measure. And I just want to say one last thing here is that the real good evaluation in protein function prediction perhaps is still an open question. So these metrics are just our best take right now. But that presents another challenge, whether we are doing this well. And how biologically relevant this actually is. So thanks.
Yeah, thank you very much. May I ask you another question? It's about the data and the labeling. So when I looked on the data, it seems to me that the labels are a little bit kind of noisy, because if I take very similar proteins, I would expect them to have sometimes exactly same labels, but I don't see it. The difference is quite big. I think it's just because, not biological reason, but just organizational, that the gene ontology doesn't have enough, I don't know, resources to make the correct labels. What do you think about that?
That's exactly correct. So the data does have some mislabels, but that may not be the biggest problem. The biggest problem is really incompleteness of the labeling. And so you can think of two levels of incompleteness. One is that some proteins are just not labeled at all, down certain branches in gene ontology, and it's just missing data. And we do our best. We have to treat it sort of as negatives when we evaluate, but right now it's very difficult to do any better. It's possible, but it's super complicated. And there's another level of this, is that sometimes the specificity of function is not well addressed. For example, some experiments will label up to depth, let's say 5, but really the experiment did more, and maybe depth 6 or 7 should have been labeled, but maybe biocurators were not confident that this was indeed the case. And so even down the branch that is correct, sometimes it's not the most specific. And so this level of incompleteness of annotations is quite interesting for the machine learning community. I'm pretty sure it exists in other types of problems, but in biological problems, it's so prevalent.
Okay. So do you see? Yeah, yes.
Sorry, about how many missed values we have?
We do not know that. There's ways to estimate this, but I think, even the algorithms that can make some kind of estimates are in development and have their own problems. So I don't think we really know truly what is the level of incompleteness in these data sets.
Can I also ask a very basic question? Sorry, I'm not that well familiar with challenge again. Is it right that we are here predicting only molecular function subset of Go terms? Okay, but even there, if I remember correctly, there are some functions that are sort of species specific, or at least family specific. So those are also included, right? But we do not. But do we have labels for proteins from what organism they come, or don't we?
Yeah, we have.
Okay, in this case, yeah, yeah, okay. This answers my question. Thank you.
Does anybody use them?
So it would be wise to use some of those, because you would not expect certain types of functions, let's say in bacterial proteins, as you can see in maybe human proteins. And so not everything that you transfer from one group to another would be relevant. And there's, again, some functions that will transfer pretty well. So it's a very tricky question, like whether some protein is an enzyme, right, that can be more easily transferred, whether maybe, whether a gene is related to speech, or maybe, tails if you're a mammal or something. If you're a bacterium, you're not going to speak. So there's a lot of things going on here.
Yes, and by extension, where can we maybe have some kind of overview of the curation methods that are used to actually assemble Go terms library? Because obviously it kind of instructs what do we have in our dataset. And I'm specifically interested in how some maybe inferred from biological logic functions can be assigned. Is there maybe somewhere an explicit description of vaporization process.
Yeah, Rauch has just put a paper there. There is also a book published in 2017 called Gene Ontology Handbook that has a bunch of chapters. And I would recommend that as well. And so I'll try to find the link to the Gene Ontology Handbook and post it here.
Yeah, thank you very much. By the way, yeah, may I kind of also, so yeah, there is some problem, yes, that different experiment, I mean, for the same property, if one laboratory makes experiment, another laboratory, they can obtain a little bit different results because we have different, I don't know, even cell lines or kind of, so is there anything which kind of gives us something like a confidence that this label kind of people are more confined that this is assigned correctly, but this label is maybe some people can hesitate or kind of there are some, you know, is there something like that in the gene ontology?
You know, I'm not aware of. I wonder whether anybody else on the call knows this information. I think there's pretty wide belief in the community that not all labels are equally good. For example, certain evidence codes, such as inferred from direct assay, is better than maybe IDA. Evidence code is maybe better than some other evidence codes, like from the expression pattern. I forget exactly what's the evidence score for that, or for genetic interactions. And so I do not know if it has been studied formally. So that's a good question. And if you can, you know, develop a system that estimates that and then use that information, I think you might get somewhere as well, you know, by how you train your models, et cetera, et cetera. But all of that said, I still feel that the bigger problem is the lack of specificity than the actual noise in the experiment. I think they take pretty good care of those, although there are errors.
But why do we have this, you say, lack of specificity? Because the proteins, we have many similar proteins. If you go down with the threshold on similarity on, I don't know, 30%, so you will have one protein covers a very huge neighborhood around it. So we can kind of think that the labels are transferring the labels to this large neighborhood. So I think my feeling that this hasn't been done by gene ontology team, and but it's possible to do. The only thing that we will not have an experimental check. Okay, colleagues, any other questions, suggestions, or?
So I just want to make one other, oh, go ahead, sorry.
No, sorry, you can go, I already asked a bunch.
Yeah, I want to just mention one thing about, you know, the relationship between sequence identity or sequence similarity and function. It's pretty complex. You know, by and large, it is true that more similar proteins will have more similar function, but sometimes key mutations can change this function quite significantly. And the overall measure on sequence identity or sequence similarity may not be the best indicator, in fact. And so between species, sometimes those key differences actually play roles. And that's why you cannot just use that, you know, BLAST to infer protein function. It's more than just that. And there's plenty of examples where key variants actually change a protein. Some, in fact, lose enzymatic activity just based on one key mutation, and then the protein does something else in a different species, whereas in some other, it's still an enzyme and something else. And so that's where it's kind of a rich type of landscape in which small changes can influence large changes in function. And that's where this problem becomes super complex.
Is there some kind of story or what you said that some small changes can, just examples or something?
Yeah, there are examples. I don't have them here by hand, but I've seen plenty of those. And so Janet Thornton used to study those. And she had even the like evolutionary tree of enzymes, how they evolve in different species, and how some kind of, I saw her talk several years ago. And I know some other examples in bacteria where that happens actually. I don't have it here with me.
Yes,
okay.
So just to reiterate, obviously functions are dependent on the environment, and our proteins in the same species are its environments. So you may want probably not only to consider a given protein, but also perhaps known, at least PPI interactors, if there are any, or something like that. Yeah. Also, I just want to say that returning to the previous question about different sources that are used to assign Go terms, it may be useful to somehow, well, first investigate how exactly they are assigned and in what proportions Go terms come from different methods, but also perhaps to use data that is conceptually similar with the Go term. Like, so what I mean is that there are some Go terms that can be inferred almost purely from sequence, but there are also others that are mostly inferred from like much higher level features. And also, I think there are some Go terms that are somewhat informed, like from evolutionary methods, from orthology, and it may be interesting to somehow weigh different embeddings for different types of Go terms.
Okay.
I'm gonna give a comment on this, that certain functions are sort of more closer to biochemistry, such as molecular function ontology will tell you whether a protein, let's say, is a particular type of enzyme, let's say kinase and etc., can be quite precise and specific, whereas biological process has something like signaling, right? That's a term, and that's a fairly broad and abstract concept, what actually signaling or developmental process, etc., may mean. And for those types of more abstract concepts, I think it's more important to use good systems data, such as protein-protein interaction networks, gene expression data, wherever there's possibility, because I feel like these types of functions will be less dependent on sequence itself, but rather on who they interact with and how they form these communities or pathway, in which pathways they participate to carry out their function. And so different types of data, I think, will play different roles in these ontologies. And again, that's a part of why this is a difficult thing. So I would expect actually every group to have slightly different model for each of these ontologies, and then hope that somehow the average of the three turns out being good.
Thank you very much. Colleagues, any other questions or suggestions, comments?
Sorry, we cannot go through some public notebook and look through why they use this embedding and how they build it.
Luda, I think we can do it on the other meeting. Maybe it will overload the today meeting. Maybe some next meeting.
Yeah, and also just one last question. Again, just with myself, but
Dean, sorry, I can't hear you very well. Can you repeat, please? Denise.
Yeah, to get information from first hands. Sorry. Is it a bit better right now? Is it better?
Yeah, now it's better.
I just wanted to ask, what about UniPark? What about proteins that are not curated? Here are only curated proteins.
So as far as I understand, maybe Predrag will correct me. So the labels which we have now, they are kind of experimentally checked. That I think includes that there was some curation process, as far as I understand.
Yes, that is definitely the case. And biocurators are also behind the literature, so they try to keep up, but it's not super easy for them to do so. There's certainly some number of proteins that are in the literature but not yet curated, didn't make it to the list.
There is a question in the chat from Max Buckley. What level of data representation would you suggest for the cellular component ontology? If molecular function is a sequence and biological process is system level, for example, protein interaction. So as far as the question is about cellular component, what you suggest as
I think it's both, actually. I feel like this one might be the hardest for a couple of reasons. And I really do not know if this is the case. But there's sometimes there's going to be a sequence signal that a particular protein goes to a particular cellular component, and then it's going to be there and do its function. So it could be some kind of substring or kind of motif in the sequence. Then you can also infer from the interactors of that protein, whether it's going to be in maybe in the cytoplasm or in the nucleus, etc. And so it seems like it's both. And also, I guess I can add that the cellular component ontology, in my mind, is the most complex and the least well thought out, or however, maybe that's too strong a term. But the number of terms in the ontology that have multiple parents is, I believe, the largest per term compared to BPO and MFO. MFO is almost like a tree and it's easier to predict on, whereas the other two are more difficult, and there's some dependencies that are quite complicated there. But I might stand corrected if these things are changing in time. Last time we checked, it's been pretty hard to understand all the levels of cellular component ontology. Rob has much more knowledge about that. If he wants to speak, I would encourage that.
Robert, if it's convenient for you to make some comments, you are very, very welcome.
Yeah, I don't have any useful comments to make because I don't know the answer. So it's an open question also for us.
Okay, thank you very much. Colleagues, any other questions, comments? Okay, there is in the chat. Historically, the biological process ontology has been harder to predict than the cellular component and the molecular function. Is it still true for today for our models? Yes.
Based on the absolute measures, that is true, but based on the relative measures compared to, say, the naive model, a cellular component, as far as I remember, and we had a slide today in the first slide, in the first presentation, I believe the cellular component is the smallest difference from the baseline model. And that's why I think it's much harder to predict than biological process. And yeah, this one. See how the cellular component, if you look at the red line, that in all the way to the right, that would be cellular component. So we are barely over that 0.55 or somewhere there, right? Whereas in biological process, absolutely you get a smaller number, about 0.4, but the red naive model is below 0.3. And so that's what I was referring to. Molecular function has the best prediction accuracy. And actually, I should also say that up to CAFA 3, in CAFA 2, I believe no model was able to beat the naive in cellular component. And so this is quite difficult.
Okay, thank you. Very interesting. Yeah. Colleagues, any other questions, suggestions, comments?
Hello, can I ask something?
Yes, sure. Yes.
Okay, so I'm new to this competition, so sorry if this is a really basic question, but when we are working with these embeddings, right? They are computed based on the amino acid sequence, right? And what do you think about incorporating the knowledge from the molecular level? So, for example, the stereochemistry. But that I mean, for example, if we taken the molecular the molecular level sequence of this, and taken, for example, some fingerprints of it. Okay, so we have some features based on the molecular level. Do you know any experiments with something like this?
Predrag,
I'm not aware of that. So it's possible that, people have used physicochemical properties for a long time to provide feature representations. And those are kind of correlated, I believe. But in many cases, we don't have structures of these proteins, so it was pretty difficult. Now with AlphaFold, we can have structures to a good extent of these proteins. So it might be more possible to include sort of more sophisticated representations.
Okay, thank you.
Thank you. Any other questions, suggestions, comments? Denise, do you have a question?
I do have a bunch, but I mean, they're kind of specific. But I just wonder about some priors in the distribution of Go terms for proteins in this large, I guess, multi-species dataset. Because I think that if you just use this species classifier that basically discards Go terms that are not applicable to the source organism, I think you already probably can get about 0.2, I don't know. I'm just interested about if somebody did this.
What organism? I didn't hear well. What organisms?
So if I understand correctly, here we do have, sorry, again, I'm not familiar with challenge. Am I right that here we do have different organisms and
yes.
probably from different kingdoms even?
Yes.
So some Go terms will be applicable only for a particular clade. And if we just use, like, without any information about the protein, just information about the source organism, like, it's interesting what would be the base level for that.
I think it will not be much because not so many terms are organisms dependent. I don't know. Predrag maybe.
Maybe 0.2 is much higher than it is actually.
I agree that it wouldn't be very a great model, but definitely that's an idea that's worth sort of incorporating in a, let's say, sophisticated prediction algorithm. Again, you don't want to predict, multicellular type of functions on a, on a yeast, or on a bacterium, on a single cellular organism. So all of that plays a role, and also different terms are have different priors in different species in the first place.
Yeah, but on the other hand, models, Denis, they can learn it by themselves. So we have training data, and I think some simple dependencies any model will see by itself. If, for example, it sees that multicellular properties are not present for this type of sequences, it will not predict.
Yes, sure, it should. At least if you have sufficient data, but
we have more than 100,000, 140,000 in the train.
I think that this still can be not enough, but actually, because I do not have a lot of compute, so would not be able to train a large language model on all the protein sequences, right? So I will have to do something much more simpler and heuristic, and maybe combine existing trained models.
Okay, Dana Marie, you have some questions, suggestions, comments, so please welcome. Colleagues, any other questions, suggestions, whatever?
Thank you very much for this just awesome discussion. I'm also new to this topic, and I was just wondering if there are some articles, like observation articles, just about this competition. So like, I'm sorry, probably I forgot the name of the lecturer. Maria, probably, right?
Liza.
Liza, I'm sorry. So like Liza told us about some methods. Maybe there are some additional materials where some people tell about methods used to solve this competition.
Predrag, do you want to comment?
Yeah, I have to say that I am not aware of any review papers in the last maybe 5 years or so. If there are, just missed them somehow. And the last one that kind of reviews methods and gives you sort of synopsis of the field was in 2016, that one review that I can point out, but that's not really that new. So I would say the best way is to use maybe CAFA papers, see those well-performing methods, such as Robert's method and Ju lab that did really well, and Orengo lab, etc., and see who they cite. And in a couple of steps, you will have pretty good understanding of the literature. And I think CAFA is there to sort of just provide the even playing field for everybody, and that we can really see how well these methods do. So we try to do this experiment every 3 years. But yeah, concrete answer, I really don't know. I think somebody needs to write a review.
Oh, okay, thank you very much.
Sure.
Thank you very much. Any other questions, suggestions? Okay, so Denis, you have some question?
I mean, my only question is it even realistic to do good prediction without a lot of compute? Because it seems like there is so much data that can be incorporated in a sort of single foundation model, like even like a large knowledge graph combined with sequence embeddings. But obviously, it will require a lot of computational resources to train this. And even though there are some disjoint models that are already trained and open source, I think there are not there are no those kind of multimodal things yet, at least open source.
Well, I think the nice thing that recent years appeared these protein language models, which are pre-trained on the, I don't know, millions, hundreds of millions of the proteins. And so they somehow incorporate the structural information, like, you know, the embeddings in the natural language processing. Yes, they work quite well, BERT or something. So we have these models for the CAFA 5, and as far as I understand, they were not available on the previous CAFA. And so now if you use these embeddings, you can then use standard machine learning, just the, I don't know, whatever you want, from linear regression to some multi-layer perceptron, and try to just forget about biology and train the models. And it's not that bad.
Yeah, this is definitely interesting. Although it probably tells us more about the embeddings that those kind of models have learned. I don't know if this is sufficient to crack the actual challenge, because obviously language models are not by default they are not known for their reasoning capabilities exactly, and even though they do have obviously some reasoning capabilities, probably those would not be enough to generalize some complex functional descriptors of a protein, because it's very logical. I think like a single mutation can change everything. But yeah, it is interesting just to investigate those embeddings, I guess.
Yeah, so the challenge will show what models are better. I know, Predrag, did you have some comments?
Nothing additional.
Okay, colleagues, so everybody is very welcome. Thank you very much for participating. Thank you very much, Liza, for making this nice presentation, and especially Predrag for your comments, and Robert, who already left, but thank you very much. So I hope we can continue these meetings, maybe, I don't know, some week later. And if you have some suggestions to make a talk, or you know some people who can make a presentation to share an experience, or just some kind of informal discussion, you're very welcome to notify me, for example, or other colleagues, and we will organize. And okay, so thank you very much again. If someone has some last questions or comments, you're welcome. If not, then have a good evening.
Sorry, can you explain where Redgard finds this big information that he has?
Sorry.
Redgard can answer
Robert.
a big question. Where he finds this information?
Robert, you mean Robert? Yes, but he is the expert. He is kind of doing this. So this CAFA challenge is not the first challenge. It's going for several years, for more than 10 years. So, and Robert is an expert in this field. As far as he participated in the previous challenges, he wrote some papers on that. So of course he is a kind of expert, like some other people.
And Predrag is too expert.
Yeah, Predrag is from the.
I hope.
Predrag is one of the organizers, so of course he is an expert.
So I can tell you about Robert. Robert has, I think he worked for his postdoc with Michael Ashburner, who developed gene ontology, who was the lead of the developer of gene ontology. So he has long history of being in this field, at least at some level, and as of late has developed several excellent methods. Our group has transitioned into organizing role because you cannot both participate and organize. And we think this is such an important problem that only as a community we can solve. And so that's why we are posing this challenge on Kaggle, and Kaggle was kind enough to fund it. It's a very important problem. And so it takes some time to really understand it, but I think sometimes you will get good ideas from so-called non-experts who will infuse something new into this whole thing, and it might just work. So we are open to seeing what happens.
Thank you very much. Yeah, it's a very interesting challenge. Okay, colleagues, any other questions, suggestions? Okay, if no, then thank you very much, everybody, and have a good evening. So the video record will be on YouTube, I guess, tomorrow, and I'll post a link on the Kaggle forum. You can see it if you need. Okay, so thank you very much, everybody.